All right. We are recording. Yay. Okay. So to summarize, a cell is the smallest thing that can be considered to be alive. And as you saw in the animation, all parts are working together toward common goals. All cells have four things in common. They all have a membrane surrounding them. We call this the cell membrane because it surrounds the cell, or the plasma membrane. It's made of phospholipids. Hopefully you remember from chapter 3 that phospholipids are a kind of lipid. They have a polar head and two nonpolar tails. Every cell has fluid inside it, and we call that the cytoplasm. Sometimes you will see it called the cytosol, but we use the term cytoplasm in this class. All cells have DNA. The way the DNA is packaged is different from cell to cell. It can be either called a nucleus or a nucleoid area. These are the best pictures I could find. So over here, the nucleus is DNA surrounded by a membrane. So we have the cell membrane keeping the cell in. And then within that membrane, there's another membrane called the nucleoid membrane, or no, the nuclear membrane. And the DNA is contained within the nuclear membrane. Whereas in the simpler cells, there is no nuclear membrane. The DNA just kind of floats around. And we call that area where it's floating the nucleoid area. And the reason that I have put a picture of a swimming pool here is because I like to think of cells as swimming pools. You know, when you fill up a pool, especially a soft-sided pool like this one, you've got the liquid that's giving it structure, and then you throw toys and people and, floaties and all kinds of things can go into the pool, and they all float in the liquid. That is exactly what a cell is like. We have something surrounding it, and then we have fluid, and then we have stuff floating in the fluid. Anyway, all cells also have ribosomes. Ribosomes are proteins that make proteins. Here is our first clicker question, and I am waiting for 6 answers. You feel free to look back at the notes. What is a cell membrane primarily made of? Ooh, there's 8 people here now. Excellent. Okay, 80% said A, which is the correct answer. 20%, 83%, 86%. Wow. Okay, 86% said A. That's good. 14% said D, cytoplasm. Cytoplasm is the fluid that is inside the cell. It does not compose the cell membrane. Here is a true or false question. A nucleus is surrounded by a membrane, but a nucleoid area is not. Is this true or false? Excellent. 100% chose true. That is correct. Here is another true or false question. A nucleus is in a prokaryotic cell and a nucleoid area is in a eukaryotic cell. True or false?
False.
All right, excellent. 83% chose false. Very good. A nucleus is actually in a eukaryotic cell, and a nucleoid area is in a prokaryotic cell. Here's a multiple choice clicker question. Remember, there are 4 things that all cells have. Which of the following is not one of them?
And they were not.
We're going to go ahead and answer in the Socrative app. Interesting. We've got some widespread here. Wow. Okay, yeah, this clicker question is a little difficult because it requires you to think back a few slides, whereas typically I give it to you right after the slide I'm talking about. So 25% said A, 25% said C, and 50% said D. At least nobody said B. That's good. The correct answer here is actually, excuse me. The correct answer here is A. So 25% of you got it correct. The cell membrane, the cytoplasm, and the ribosomes are 3 of the 4 things that all cells have. All cells also have DNA. So let's go back up to all cells have a membrane, a cytoplasm, DNA, and ribosomes. All right, we have 2 main classes of cells, but first let's talk about a little bit of vocabulary. The word organelle means little organ. Organelle, L means small. So it's a structure that performs a specific specialized function within a cell. If you think about organs, you have the stomach, which does a specific specialized function. You have the liver, which does a specific specialized function. You have the brain, all these organs. Cells are similar, but we call them organelles instead of organs because they're so small. There are 2 types of organelles. Ones that are surrounded by membranes, we call those membrane-bound organelles, and ones that are not surrounded by membranes, and we call those non-membrane-bound organelles. So membrane-bound organelles, I just want to show you, here's a couple. This is the mitochondria, and see that there's a membrane surrounding it. This is the endoplasmic reticulum. There's membranes surrounding those. This is a ribosome. It is a non-membrane bound organelle. And these are the centrioles. They are also non-membrane bound. Keep in mind, all membranes are made of a phospholipid bilayer. Here I've got a phospholipid shown for you. It's polar hydrophilic head and the nonpolar hydrophobic tails. And then we've got here our phospholipid bilayer, 2 layers of phospholipids. Remember, they naturally form this shape because the polar hydrophilic heads are trying to protect the nonpolar hydrophobic tails from the surrounding environment. Cell membranes are made of a phospholipid bilayer as well. There's many different kinds of membranes. Here's our next multiple choice question. Go ahead and answer in Socrative. Excellent. 86% chose the correct answer, which is B, a phospholipid bilayer, including the cell membrane, which is sometimes called the plasma membrane. Here I've got a picture of a cell membrane. We went over this a little bit in chapter 3. We've got phospholipids, as you can see here. We've got the little heads and the little tails making the phospholipid bilayer. The cell membrane also has proteins stuck in it. That's these purple blobs. We have carbohydrates, which are these green tails coming out. And we have cholesterol, which are the yellow molecules that are sticking between the phospholipids right there. You know, I think I can turn this into a laser pointer. There we go. That's more like it. Okay. The first type of cell is a prokaryotic cell. Pro means before, prokaryotic. There's no nucleus. Instead, we have a nucleoid area. The nucleoid area is this area right here where the DNA is just kind of generally floating. They have one single circular chromosome. A chromosome is a piece of DNA. Humans have multiple linear chromosomes. We are not prokaryotic, but all prokaryotic cells only have one single circular chromosome. One of the distinguishing features is that there are no membrane-bound organelles, meaning they do not have a nucleus because a nucleus is considered a membrane-bound organelle. And all prokaryotic cells are called prokaryotes. These images are bacteria. Bacteria are prokaryotes. Here's our next clicker question. God, I love technology. imagine back like in the 1818 Spanish flu when they didn't have technology and everybody was confined to their house for like a couple of years? Everyone just fell back 2 grades in school. And snow days, well, maybe they didn't care about people getting cold. I don't know. 83% chose A, which is the correct answer. Very good. So we have prokaryotic cells, and now we also have eukaryotic cells. The prefix "eu" means good. These guys have a nucleus, and they have multiple linear chromosomes. Remember, chromosomes are just pieces of DNA. So instead of a single circular chromosome, eukaryotic cells have multiple linear chromosomes. They are larger, they have membrane-bound organelles, and we call them eukaryotes. Plant and animal cells are eukaryotes. Take a look at this. This is a eukaryotic cell. We have the nucleus here. You can see how it's divided into smaller packages, whereas the prokaryotic cell, everything's just kind of floating around. But the eukaryotic cell, we have all these little organelles doing their various jobs. Here is our next clicker question. I hope everybody's enjoying your snow day. My sister lives in San Antonio, and she said that it's supposed to be below freezing tomorrow. So all of the schools are closed. There's not even any snow in the forecast. Maybe they're afraid of becoming icicles or something. All right, 83% said C. C is the correct answer. Very good. Now, what is true of prokaryotic cells? Okay, 67% of you chose the correct answer, which is B. They have a single circular chromosome. 14% chose A. Actually, prokaryotic cells are smaller. And 13% chose C. They do not have DNA. DNA, remember, is something that all cells have. It's one of the 4 things that all cells have. Here is another one. It's the same question phrased differently. Let's see how many people get it. Excellent. Everybody chose A. Good job, y'all. All right. Just a sec. God. Okay, I have these neighbors that I'm pretty sure one of them is bipolar, and they're constantly screaming and banging on things. And I'm never sure whether they're banging on my door or on the wooden railing outside. But I mean, they're crazy. I don't want to answer the door anyway. Okay, so now I'm going to show you another video of a white blood cell chasing a bacterium. Remember that many parts of the cell work together towards a common goal. So I want you to watch for 3 things. The white blood cell is going to change shape, and the prokaryotic cell, the bacterium, is much smaller than the eukaryotic cell. And the white blood cell is following the chemical scent of the bacterium. That is how it knows where to go, because as the bacterium moves along, it leaves behind it a little trail of chemicals. There is no sound to accompany this video. So there's the white blood cell. All these other guys are red blood cells. And just notice how tiny the bacterium is. The prokaryote is much smaller than the eukaryote. Ah, he got him. Yes. And the reason that the eukaryotic cell is able to chase the prokaryotic cells because it can change shape, because all of the parts in the cell are working together to chase that bacterium. Let's watch this one. This is inside a eukaryotic cell. What? No. Okay, well, I guess we're not going to watch that. Moving on, recall that eukaryotes, they have all the organelles. Prokaryotes only have non-membrane bound organelles. Let me get my laser pointer back. So eukaryotes have all the organelles, whether they're membrane bound or not. But prokaryotes do not have membrane bound organelles. Excuse me. Everything in a cell is organized around creating proteins. They do other stuff too, but proteins is the main point of every cell. Right here, I've got a muscle cell. You can see the muscle fibers running from left to right. And then within these are mitochondria. Mitochondria, remember, produce ATP. They are the powerhouse of the cell, and your muscle cells need a lot of power. So that's why there's so many mitochondria here. Those are also muscle cells down there. All right. From here on out, we're only going to be talking about eukaryotic cells. These are the two kinds of eukaryotic cells. We have animal cells and we have plant cells. I want you to notice there's three main differences between plant and animal cells. First of all, the plant cell has this rigid cell wall surrounding it, whereas the animal cell does not. Also note, there is a plasma membrane or a cell membrane surrounding both cells. It's just that the plant cell, in addition to the cell membrane, has a cell wall. Second difference, plant cells have chloroplasts. Animal cells do not. Third difference, plant cells have this thing called a central vacuole. We'll learn about that in a minute. And animal cells do not have central vacuoles. So now we're just going to go through the structures of the cell and their functions. The plasma membrane, also called the cell membrane, forms the boundary of the cell. Remember, it's made of a phospholipid bilayer, and it is selectively permeable. It means that some things can go in it and some things can go through, others not. Just like a coffee filter. A coffee filter is a selectively permeable membrane. The coffee cannot pass through it, but the water can. And this is really important for the cell because we don't want everything to be able to get in and out of a cell. Some stuff needs to stay in and other stuff needs to stay out. The cell membrane has a lot of proteins stuck in it, and the proteins are used for communication, and they're also used as gates to control what goes in or out of a cell. We'll talk more about this in chapter 5. The fluid mosaic model of, what is this? Okay, the fluid mosaic model is a model that we use to explain the movement of the cell membrane. The cell membrane, nothing in a cell is stable. It's always moving. There's always activity happening. What? There we go. Are you fucking kidding me? Are you fucking kidding me? No! Wait, maybe the rest of it. Oh, yeah. The rest of it's behind the. Okay. I'm a little technologically illiterate. All right.
Between the living machinery of the inner cell and the harsh conditions of the outside world stands the cell's plasma membrane. As crucial as this barrier is, it's surprisingly flexible. Push it and watch it move. Poke hard enough and it might break and begin to regroup. The lipid molecules of the membrane naturally assemble in a double layer because their tails repel water as their heads attract it. Throw in some cholesterol and a few carbohydrates, and you have the basic structure of a plasma membrane. Within these lipid molecules, we also find different proteins which do various things for the cell. For instance, They receive signals from the world outside, or they transport nutrients and waste. So nature composes the membrane with a combination or mosaic of different lipids, carbohydrates, and proteins. And these molecules are not stationary. They constantly move within the structure, fluidly changing their positions. The survival of all life.
Blah, blah, blah. The survival of all life depends on the fluid mosaic model. Actually, it does. I shouldn't say blah, blah, blah. But what I want you to notice is that this phospholipid bilayer is moving fluidly. Nothing is stable. And it is a mosaic of different protein, different kinds of molecules. We've got proteins, carbohydrates, phospholipids, lipids, other kinds of lipids. Stop that. Okay. All right. Here is another picture. I just wanted you to have this for visual reference whenever you are studying. This is a cell membrane, which remember is called a plasma membrane sometimes. It's a mosaic, and these are the things that you can find in it. Here is our next clicker question. It is a short answer, and I'm waiting for 7 answers. I'm really impressed with our turnout today. Yesterday I had like 3 students out of 23 show up. Excellent. Lipids, phospholipid bilayer, proteins, lipids, and proteins. I do see the answer plasma. That is an interesting one. Okay, because we call it the plasma membrane does not mean it's made of plasma. Plasma is a state of matter that is not liquid, solid, or gas. And I guess it's also what's in your TV. But plasma is not a thing that is in the cell membrane. It is called a plasma membrane. I'm not sure why it's called the plasma membrane, actually. Wish I could explain that to you. All right, so that's the cell membrane. Secondly, we've got the cytoplasm, this fluid that's inside the cell. It's also called the cytosol, but we're going to use the word cytoplasm here. Next, we have the nucleus. We are not discussing the nucleoid area because we are only talking about eukaryotic cells here. Recall, nucleoid area is in bacteria only and prokaryotic cells only. Let me turn this into a laser pointer again. So a nucleus is DNA surrounded by a membrane. You can see here we have the nuclear membrane, and there's a cutaway revealing the DNA inside. The nuclear membrane is sometimes called the nuclear envelope. Remember, hopefully you remember from chapter 3, the nucleus does not control the cell. The DNA that is within the nucleus controls the cell. So you can call this the brain of the cell, but it's actually the DNA that does the controlling of the cell. And remember, a nucleus is only in eukaryotes. There's the nucleoid area, which is in prokaryotes. Nucleolus is within the nucleus. It is a dense knot of DNA. You can see it in this image of an animal cell. We've got the nuclear membrane surrounding the nucleus. We've got the DNA, and then there's a dense knot of DNA inside, and that is where ribosomes are made. Recall that ribosomes are proteins that make proteins. All right, this is one of my pet peeves right here. Nucleus, Nucleus, that is a word. Nucleolus, Nucleolus, that is a word. Nucleus, Nucleous, this OUS, that's not a thing. There's no such thing as a nucleus. Please do not ever write that. There is no nucleus. Yes, spelling counts in college, and this one counts more than all the other spellings. All right. What is made the nucleolus? All right, 100% of you chose D, ribosomes. Very good. Speaking of which, let's talk about ribosomes. Ribosomes are proteins that make other proteins. They are one of the four things that all cells have. There's two places they can be found within a cell. They can be found free, just roaming about in the cytoplasm, or they could be found attached to what's called the rough endoplasmic reticulum, which we'll talk about next. All cells have ribosomes. Which is true of ribosomes? Oh, we lost two people. There's only 5 students now. All right. 100% chose B, proteins that make proteins. Very good. That is the correct answer. Next, the endoplasmic reticulum. That's this sort of maze-like structure of membranes that runs throughout the cell. This kind that kind of looks like funnel cake is called the smooth endoplasmic reticulum. And the rest of it that looks like a maze is called the rough endoplasmic reticulum. And the smooth and the rough have different functions. And I want you to notice that they're attached to the nucleus right there. The rough endoplasmic reticulum has ribosomes attached to the outside. You can see in this cutaway here, you've got some dots attached to the rough endoplasmic reticulum, whereas the smooth endoplasmic reticulum does not have those ribosomes. The function of the rough ER, the darker purple here, is to take the proteins that are made by the ribosomes on its surface. So we've got these ribosomes on the surface of the rough ER that are making proteins. They spin those proteins right down into the rough ER and then modify them and send them to the next step of the protein making process, which is the Golgi body, which we'll talk about next. The smooth ER, on the other hand, this lighter purple here, has no ribosomes, and that's where lipids are made. We've got hormones, cholesterol, phospholipids, steroids. Those are all made within the smooth ER. I've got a picture here. This is a cross section of a microscope picture. You can see that the rough endoplasmic reticulum has all these dots, these ribosomes attached to the outside. And the smooth endoplasmic reticulum has no dots or no ribosomes. Here's our next clicker question. Okay, 83% chose C, which is the correct answer. Rough ER has ribosomes on its surface. 14% chose A. A is incorrect because it's actually smooth ER that does not have ribosomes on its surface. It has a smooth appearance. That's why it's called smooth ER. Here's a true or false. All right, 83% chose false, which is the correct answer. The function of the rough ER actually is to create proteins or take created proteins, modify them, and send them to the Golgi body. Ooh, mitochondria, the powerhouse of the cell. Singular is mitochondrion, not mitochondria. Yeah. Me, what are taxes and how do I pay them? School system, worry not. Mitochondria is the powerhouse of the cell. That is unfortunately quite true. This is where ADP is made into ATP, and we've watched this animation before. We're going to watch it again. Science says that you need to see something at least 3 times in order to remember it. So this is the second time. Okay. Well, where's the volume? Taxes.
Phosphate groups that releases energy. Let's take a look at how exactly this happens in the cell. Here we have a process, and as you can see, it's in desperate need of some energy. Fortunately, there's ATP that comes along, and when that ATP comes to the process, it breaks the bond between 2 phosphate groups and donates 1 phosphate to that process or that protein or whatever it is that needs that energy. When this happens, the process gets the energy that it needs, and it can then do whatever task it needs to accomplish. Let's get rid of this process for now. And now you can see we are left with 2 phosphate groups, so it's no longer ATP, it's ADP, or adenosine diphosphate. If we want to use this molecule again, we need to recharge it as we would do for a rechargeable battery. In order to do this in the cell, we need a very special organelle called the mitochondria. ADP then goes to the mitochondria, and the ADP gets recharged by the mitochondria adding another phosphate on, and it's now ATP again.
So the mitochondria is where ATP is made, essentially. All right, now this next video is a little bit long. We're not going to watch all of it, but it explains the theory of endosymbiosis, which is the theory of how mitochondria and chloroplasts came to be. I love Mr. Anderson.
Hi, it's Mr. Anderson, and in this podcast, I'm going to talk about one of my favorite topics in all of biology, and that's endosymbiosis. If you didn't know this, there are 2 major groups of cells. We have prokaryotic cells, and then we have eukaryotic cells. An example of a prokaryotic cell is like a bacteria. They simply have a cell membrane, cell wall. All of their DNA is organized in a nucleoid region, and they're fairly simple and fairly small. In a eukaryotic cell, we're going to have a nucleus. We're going to have organelles like endoplasmic reticulum, Golgi apparatus, mitochondria. But what we find is when we look in the fossil record, life started about 3.6 billion years ago, and we just see prokaryotic cells for the longest of times. In other words, we don't see eukaryotic cells show up until around 2 billion years ago. And it puzzled scientists how this shift was made because there are clearly 2 different evolutionary pathways. The pathway of the small prokaryotic cells and then the larger eukaryotic cells. And they eventually settled on an idea called endosymbiosis. And what does that mean? Well, let's break it down. Endo means within, symbiosis means together, and bio just means living. And so basically we have organisms that are living together within one another. So that's weird. What does that mean? Well, basically, when I read about this the first time, it puzzled me. What we think is way back in the day, we had these aerobic bacterium, ones that were doing cellular respiration, so they were breaking down food in the presence of oxygen. We also had these cyanobacterium that were doing photosynthesis, and they were essentially engulfed by another host cell, and they became the mitochondria and the chloroplasts that we have today. And so that's pretty cool. In other words, these cells became part of a cell and eventually became that cell. That means that the mitochondria that are found in all of your cells are kind of like hijackers that have been inside our cell for billions of years. Now you can see why scientists would have a hard time.
All right, that's all we're going to watch of that. So the mitochondria is called the powerhouse. Why? Next clicker question. Let me set that up. Excellent. Everybody chose B. Great answer. Hey, can everybody please mute your microphones? Thank you. Sounding a little bit like my neighbors there. Anyway, okay, vesicles. There are many types of vesicles classified based on what is in them and their function, but we're only going to talk about transport vesicles and lysosomes. Transport vesicles, as implied by their name, transport things. You can think of them as taxis. They just pick things up, take it from one place, and put it in another place. Lysosomes are also vesicles, but they pick up trash and waste, and then they digest it and expel it from the cell. They're also used to digest food. So basically, they're little sacks, little membrane-bound sacks that go around the cell and pick up waste or food and digest it. They contain digestive enzymes. You can think of the lysosomes as the garbage disposals. Let's go over here. Remember, you can always find the animations on your own. Go to student view by going down here to the module textbook animations, and we are in chapter 4, so we're going to click on chapter 4.
Like secretory proteins and some other proteins, proteins destined for lysosomes are made on ribosomes bound to the rough ER and moved through the endomembrane system. In this case, the lysosomal protein-containing vesicle that buds from the Golgi apparatus is the lysosome itself. Like secretory proteins.
No. Where's the mute? Okay. So assuming that this is a protein, this purple thing, watch that it went from the rough ER to the Golgi body, and then from the Golgi body, it goes through from pancake to pancake, layer to layer, getting more and more modified. And then it's packaged into its final transport vesicle. The Golgi apparatus, the post office. This receives newly made proteins from wherever they have been made, packages them into transport vesicles, and sends them off to their final destination. So it gets proteins from one place and sends them to another place. This should be really easy. All right, 83% chose the correct answer, which is the Golgi body. Here is a cross-section of the cell. I want you to notice the relationship of the Golgi apparatus to the endoplasmic reticulum in a cell that secretes proteins. So cells in your body that secrete proteins, there's so many different kinds, but I'm going to use the example of a nerve cell. Nerve cells create what's called neurotransmitters in the body, and they'd secrete them. And those are made in the rough endoplasmic reticulum, packaged into transport vesicles. They go to the Golgi body to be modified and finished, and then packaged into another vesicle that goes to their final destination. Now, there is one thing about this picture that I do not like, and it is that they're showing proteins right here in the smooth endoplasmic reticulum. But proteins only come from the rough endoplasmic reticulum. Smooth endoplasmic reticulum, remember, is to make hormones and cholesterol. All right, next organelle, centrioles. These are the centrioles right here. They sort of look like canisters, and they produce protein fibers, also known as spindle fibers, that drag chromosomes around during cell division. They push and pull the chromosomes until they are exactly on the opposite sides of the cell, and then the cell splits down the middle. Here is a short answer question. Whoops. What do centrioles do? I think the last clicker question had 5 answers, so I'm going to wait for 5 answers. Very good. Okay. We have produce spindle fibers, make protein fibers, produce proteins and drag chromosomes, produce spindle fibers, produce protein fibers. Ooh, 7 answers. Pull and separate chromosomes. Excellent. All very good answers. Okay, cytoskeleton. This is the network of protein fibers that runs all throughout the cell, and it has several functions. So we call it the cytoskeleton. The prefix cyte means cell, and everybody knows what a skeleton is. So in humans, our skeleton, our bones, is what gives us structure. But in cells, the cell skeleton, this network of protein fibers running throughout the cell, that is what gives the cell structure. And it can be broken down and reassembled at will. It's not stable. So remember, one of the things that it does, well, here's some more, these structures radiating out from the centrosome right there. So what it does, remember the white blood cell in the microscope video that was chasing the bacterium? It was moving and changing shape. And the reason was that the cytoskeleton was being broken down at one end and reassembled at the other end, allowing the cell to grow at one end and shrink at one end, giving the cell movement. It is also used, let's see if I have, no, I don't. Okay. It's also used as a highway system for things like vesicles to move along. And vesicles, you know, these guys, these tiny little dots here, those are vesicles. Remember, vesicles carry things. And you can see that they are walking along these protein fibers. So when a vesicle, for example, when a protein comes from the rough endoplasmic reticulum, it's packaged into a vesicle. And then the vesicle moves along these protein fibers one way and then another way until it gets to the Golgi body. All right. Cilia and flagella. These guys help with movement. Flagella and cilia are different because flagella are longer and they sort of look like whips. And you can have several at a time or just one at a time. Right here, these are sperm cells. Cilia are much shorter. They're like these finger-like projections that are all over a cell. Whenever the cell has them, there are many at a time. But both of these, not all cells have them, only cells that need to move. Let's watch this. No.
As I'm in the midst of applying for residency right now, wish me luck, Grammarly has again been a lifesaver with my application.
Many cells use flagella for moving.
Okay. All right, so here's some flagella. This is a light microscope image, so you can't see them very well, but you can sort of see them coming out one end of this tiny organism called a euglena, helping it move. Here are some more flagella. They're just coming out this end, and they're moving very, very quickly back and forth like little whips. And then here we've got some cilia at the top there. That's a rotifer. Where's our paramecium? There we go. All right, you can see the tiny little cilia completely covering this paramecium, helping it to move. Yeah. That's also pretty difficult to see because of the microscope they used. Now, all right. Here's our next clicker question. You can sit here. All right, the correct answer is A. 83% of you chose A. Cilia are shorter. Very good. 14% chose C. Both are present in all cells, but no, actually the thing is that neither of them are present in all cells. Only some cells have cilia and flagella. All right, now let's talk about specifically plant cells. Plant cells have three things that animal cells do not have. First, they have a cell wall, which provides structure and protection. It is made of cellulose. So in this image here, let me find my pointer again. In this image here, you can see the cell wall surrounding the cell, giving it some kind of structure. It's like a box. And the central vacuole is the second thing that's unique to plant cells. It's this large fluid-filled sac in the middle of the cell, and it stores water and gives structure. And it stores waste as well. So when water, like here's a picture of the importance of the central vacuole. When the cell does not have enough water, the central vacuole is shrunken like this, and that causes the sides of the cell wall to bend in slightly, which means that a plant without enough water is going to be wilty. But when there is enough water in the central vacuole swells and causes the cell wall to become straight and rigid. And that is why when you water your plant enough, it looks like this, because the cells have structure instead of being shrunk. The third thing that only plant cells have is a chloroplast, and a chloroplast uses light energy to produce glucose. That's very important. Glucose is a key term there. This is where photosynthesis happens, and it contains chlorophyll. Chlorophyll is what does photosynthesis, and we'll talk more about that in Chapter 7. The theory of endosymbiosis also applies here. Recall that theory said that mitochondria and chloroplasts used to be their own free-living organisms, but then they were engulfed by a larger cell, and the smaller cell, which would be the mitochondria or the chloroplast, made energy for the larger cell, and the larger cell provided protection for the smaller cell. This is very important. A lot do not fall prey to the common misconception that plant cells only have a cell wall. They have a cell wall, but they also have a cell membrane. The cell membrane is within the cell wall. Plant cells never have flagella or cilia. All right, here's our next clicker question. All right, 83% chose D, which is the correct answer, flagella. Here's another question. We are coming to the end here. Interesting, we're split. Okay. 17% said A, 50% said C, and 33% said D. So A is incorrect because we have mitochondria, yes, but we also have a cell wall and chloroplast. Remember, those are only found in plant cells. Nobody said B, that's good. 33% said D. That is not correct because bacterial cells, remember, are prokaryotic cells, and they do not have mitochondria, cell walls, or chloroplasts. So the correct answer here is C. Here is another one. Oh, oh goodness. Okay, 50% of you chose the correct answer, which is B, animal cell. We can tell that it's an animal cell because it has mitochondria, so we know that it is eukaryotic. That rules out answer D, prokaryotic. It has cilia, and we know that plant cells do not have cilia, so we know that it's got to be an animal cell. Bacterial cells are prokaryotic cells, so C and D are both not possible answers. What about this one? Okay, 60% said C and 40% said D. The key word here is nucleoid area. And then another key word is one circular chromosome. Those are characteristics of a prokaryotic cell. So the correct answer here is C. All right, we have reached the end of the lecture. I really recommend that you do these 4 things to review because they will help you organize the information that was found in the lecture. And that way you won't have to do them when you are prepping for the test. You will already have these things done because I can guarantee you right now, prokaryotic versus eukaryotic cells and plant versus animal cells will be on our test. These are my sources. So those of you who have hung around this long, first of all, well, where's my Teams meeting? There we go. First of all, you do not have to take the end of lecture quiz. And second of all, I want you to type your name either into the chat right here or into, I'm going to start a short answer question on Socrative. And those of you who do that will get a free 10 points on the quiz, 100% free. So go ahead and type your name in, and then you can leave.